All right, get ready, because we're diving deep today
Ooh, diving deep. I like it.
into the world of molecular biology.
Sounds complicated.
It can be
That's
because...
fascinating, right?
Totally. You sent us a ton of research
Double stack, yeah.
on post-transcriptional regulation in eukaryotes.
Okay, so
So basically, how genes are controlled after that initial DNA to RNA step.
it's like the behind-the-scenes stuff.
Yeah, it's like uncovering the secret code
Totally a secret code.
that determines how our bodies actually function.
Exactly. Way beyond that simple idea
Yeah,
that one gene just makes one protein.
right. Because we know that's not quite right.
Not even close. We're
I
talking
see.
about a whole system
Oh, wow.
of checks and balances,
Yeah.
fine-tuning,
Okay.
making sure the right proteins are made at the right time
Oh, I see.
and in the right places.
So kind of like quality control.
Exactly. Like a cellular quality control system.
I like that.
Yeah.
So one of the key players in this fine-tuning process
Yeah.
is alternative splicing.
One of the big ones, yeah.
And this is where it gets really interesting.
It does.
Because it's like our cells have
They have like, what is it?
their own editing studio
Editing
for
studio.
those genetic instructions.
It's wild.
So can you break that down for us a little?
Okay, imagine this, right?
Okay.
Your DNA, it's like this massive library
Huge library.
filled with blueprints
Oh, blueprints. I like that.
for every protein your body needs.
Okay.
But instead of using the entire blueprint every time,
Yeah.
alternative splicing allows the cell to be choosy.
Choosy.
Pick and choose, like
Yeah.
specific instructions
Oh, wow.
from within a gene,
So not the whole blueprint.
not the whole thing.
Interesting. So it's more like
It's more like what?
choosing different chapters from a book
Exactly. Chapters from a book.
to create a customized story.
You got it. Customized instructions, custom proteins.
So instead of one gene, one protein,
Yeah,
it's more like one gene, multiple protein possibilities.
that's it. One gene, multiple proteins. Yeah.
And those different proteins, they have a name, right?
They do. They're called isoforms.
Isoforms.
And they can have completely different functions.
Oh, wow.
It's amazing, really.
Do we have like a real world example of that?
A great example. The Drosophila mechoc gene.
Drosophila, that's the fruit fly, right?
The fruit fly. Yep. So this gene makes a muscle protein,
Okay.
but through alternative splicing,
Yeah.
it produces different isoforms of that protein.
So different versions?
Different versions fine-tuned for specific muscle types in the fly.
Like specialized muscle proteins.
Exactly. Specialized for each muscle type.
That's incredible. And scientists actually,
They actually was.
they swapped those isoforms in fruit flies,
They did.
and it really affected their wing movements.
They really did, yeah.
The flies with the mismatched isoform,
What happened to them?
they couldn't fly as well.
They couldn't fly as efficiently. That's right.
Wow. So even small changes,
Tiny change.
sort of a protein structure, can really change how it works.
Drastically change its function, yeah.
That's amazing. And you mentioned this earlier.
Yeah, what did I mention?
That alternative splicing is this major contributor
Huge contributor.
to all the different proteins we have.
It's a big deal.
So, like, how many different proteins can we actually make?
Well, think about this. The human DSCAM gene,
Okay.
just one gene,
Yeah.
can potentially make
Okay.
a mind-boggling 38,016 different proteins.
Wait, 38,016. That's just from one gene.
Just from one gene through alternative splicing.
That's insane. So we have roughly 20,000 genes,
About that.
but alternative splicing creates
It creates what?
hundreds of thousands of protein variants.
At least that many, probably more.
So what does that mean for how we think about our DNA?
Well, it challenges that old idea
Which one?
that our DNA is just this static blueprint.
Ah, because it's more dynamic
Way more dynamic.
and adaptable.
Exactly. Much more adaptable than we thought.
I'm starting to think of ourselves like master chefs,
Master chefs.
taking this basic recipe.
The DNA.
Yeah, and creating endless variations.
Endless variations.
Depending on what's needed.
Exactly. It's fascinating.
This whole editing process,
About it.
it can't be random.
Right. Definitely not.
There have to be some controls in place.
Oh, absolutely. There are specific sequences
Okay.
within the RNA itself.
Like what?
Things like splicing enhancers and silencers.
Enhancers and silencers.
Okay, and they act as signals, you see.
Signals for what?
They tell the cell how to splice.
I see.
And then there are these RNA binding proteins.
RNA binding proteins, those are
They're like the editors, you could say.
the editors.
They interpret those signals
I had it.
and guide the splicing machinery.
It's really complex.
Incredibly complex.
And as with any complex system,
What happens?
sometimes things can go wrong.
Ah, you're talking about when splicing goes haywire.
Exactly. What happens when there are errors?
That's when we start seeing what are called spliceopathies.
Spliceopathies. What are those?
Diseases, basically.
Diseases caused by?
Errors in splicing. Yep.
So, like, what kind of diseases?
Well, there are some really serious ones.
Okay.
Like myotonic dystrophy.
That affects the muscles.
Muscles and other tissues, too.
Oh, wow.
And then there's spinal muscular atrophy.
And that's
a neurodegenerative disease, yeah.
So these are serious conditions.
Very serious, yeah.
It's a reminder that even though this is happening...
Happening where?
At a microscopic level, inside our cells,
Right.
it really impacts our overall health.
Huge impact, absolutely.
So we've talked about how proteins are made.
We have.
And how they're edited through alternative splicing.
Right.
But once a protein is made, is that it?
Is that it? Is that the end of the story?
Yeah. Is its fate sealed?
Oh, not at all.
Or is there even more fine-tuning
Oh, there's more
happening after that initial production?
You bet there is. There's a whole other level of control.
Another level. Okay, I'm intrigued.
It's called post-translational modification.
Post-translational modification.
Yeah. It means even after a protein is made, it can still be tweaked and modified.
Tweaked and modified? What do you mean?
Well, the cell can regulate its activity even further.
So it's like the cell is saying...
It's saying what?
Okay, we've made this protein.
Got the protein, check.
But we need to make sure it's working properly.
Make sure it's working.
And doing its job at the right time and place.
Exactly. It's all about fine-tuning.
So many steps.
So many steps, yeah.
And how does that happen? What are some of the ways?
How does that happen? Oh, there are a number of ways.
Okay,
it can happen from adding or removing chemical groups.
Adding or removing?
Yeah, like little tags.
Wow.
Or even tagging the protein for destruction.
Oh, wow. For destruction.
Yep, if it's not needed anymore.
It's like a cellular recycling system.
It is. Breaking down old components
Okay.
to make room for new ones.
So efficient. But before we get into those details,
Sure.
Let's talk about another aspect of post-transcriptional regulation.
Okay, what aspect?
The lifespan of those mRNA molecules.
Yeah, mRNA lifespan, right?
How does the cell decide
when to hit the delete button?
That's where mRNA stability and degradation come in.
Stability and degradation. So, like...
It's all about how long those mRNAs hang around.
Got it.
It's crucial.
Crucial for what?
Because the amount of protein a cell makes, it depends not just on how much mRNA is made,
okay,
but also on how long those mRNAs stick around.
So, even if a gene is actively making mRNA,
right,
if that mRNA is quickly degraded,
gone,
you might not get much protein at all.
Might get nothing.
So it's like having a leaky faucet.
A leaky faucet, okay.
Even if the water's running,
right,
if it's all draining away, you're not going to fill the sink.
Exactly. That's a great analogy.
Thanks. And this leakiness.
This leakiness, yeah.
It can be controlled in various ways, right?
Oh, absolutely.
Like how?
Well, for instance, those protective caps.
Protective caps? What are those?
No,
on the ends of the mRNA molecule.
Okay.
The 5-foot cap and the 3-foot poly A tail.
Those are the caps.
Those are the caps.
Okay.
They play a big role in how long the mRNA lasts.
So if those caps are removed...
If those caps are gone...
The mRNA is basically vulnerable.
Vulnerable, yeah. Open to attack.
Open to attack by what?
Well, there are enzymes.
Enzymes are...
They're like little Pac-Man
okay.
that are designed to shorten or remove those tails.
So they eat the caps.
They chomp on those tails.
And that makes the mRNA...
That makes the mRNA what?
A target for degradation.
Exactly. A target for degradation.
By what? Cellular garbage disposals?
Well, sort of.
Sort of.
Yeah, there are complexes, like the exosome.
Exosome.
That's one of the garbage disposals.
Okay.
It degrades mRNA from one end.
And are there others?
Oh, yeah, there are enzymes like XRN1.
XRN1, okay.
That chomp away from the other end.
So it's like a multi-pronged attack.
You could say that.
But why go through all this trouble?
What trouble?
To destroy perfectly good mRNAs.
Good question.
Wouldn't it be more efficient to just keep them around?
Well, it all comes down to control.
Control.
And efficiency.
Right.
Cells need to be able to adjust protein levels.
Okay.
Quickly and precisely.
Ah, so sometimes that means...
Sometimes it means what?
Getting rid of mRNAs.
Getting rid of them.
Yeah. To prevent the production of proteins.
Proteins that are no longer needed, yeah.
It is like a cellular recycling system.
Exactly. Breaking down the old to make room for the new.
Very cool. And this process isn't random, right?
Definitely not random.
You're regulated.
Tightly regulated.
How so?
Well, there are specific sequences within the mRNA.
Within the mRNA, okay.
And then there are those RNA binding proteins again.
Ah, those RNA binding proteins.
They're everywhere.
Seems like they're involved in everything.
Pretty much, yeah.
So they can either stabilize or destabilize the molecule?
Exactly. They can either protect it or target it for destruction.
And this whole system of mRNA degradation,
It's pretty important.
I bet. In what way?
Well, think about cancer research, for instance.
Cancer research, okay.
There's a protein called TTP.
TTP.
It promotes mRNA degradation.
So it helps get rid of mRNA.
Exactly. But in many cancer cells,
What happens?
TTP is downregulated.
Downregulated meaning?
Meaning it's not working as well.
Oh, I see.
So those mRNAs that should be degraded,
They stick around.
they hang around, yeah, and they keep making proteins.
Proteins that could be driving tumor growth?
Exactly. Proteins that fuel tumor growth.
So if researchers could boost TTP activity,
What could happen?
they might be able to slow down tumor growth.
That's the hope. It's a potential target for therapy.
Wow. So this mRNA degradation process,
Super important.
crucial for so many things.
Absolutely. Maintaining normal cell function, preventing disease.
But it's not just about how long mRNAs stick around, is it?
What else is there?
It's also about making sure that the mRNAs that are translated,
And ones that are actually used to make proteins.
yeah, that they actually make functional proteins.
Right, because you don't want faulty proteins running around.
So there's a quality control system in place for that too.
Absolutely. One of the most important mechanisms.
Tell me about it.
It's called nonsense-mediated decay,
Nonsense-mediated decay.
or NMD for short.
NMD. Got it. What does it do?
Think of it like a cellular spell checker.
A spell checker.
Yep. It identifies mRNAs with errors.
Errors like typos.
Kinda, yeah. Like a premature stop codon.
Okay.
And it prevents them from being translated into faulty proteins.
Faulty proteins, so those would be...
Those would be like a misfolded protein or a truncated protein.
And those could cause problems.
Definitely. Imagine a protein as a complex machine.
Okay.
If a key part is missing or messed up.
The whole machine malfunctions.
Exactly. And that could be really bad for the cell.
So NMD steps in to prevent that.
That's right. It recognizes the errors
Okay.
and triggers the degradation of the faulty mRNA.
Ah, so it sends it to the recycling bin.
Yep. Prevents those potentially harmful proteins from being made.
That's amazing. Our cells are so smart.
They really are. They have all these systems in place.
To keep things running smoothly.
Exactly. It's all about maintaining balance and order.
It's really impressive.
It is. And we're still learning so much about how it all works.
Well, I have one more question before we move on.
Okay, shoot.
If mRNAs are the messengers, right,
Yeah.
and proteins are the final product,
Right.
are proteins just passively carrying out their functions?
Passively. Oh, that's a good question.
Or is there another layer of control?
There's always another layer.
Even after they're made?
You're thinking ahead.
Yeah.
And the answer might surprise you.
Okay, lay it on me.
Even after a protein is synthesized, it can be further modified and regulated.
Wait, so even after all that,
Even after all that.
the splicing, the degradation, the quality control,
Yep, all of it.
there's still more fine-tuning happening.
You got it. The cell really wants to get things right.
It's obsessed with perfection.
You can say that. And this level of control,
It's got to be important.
it's crucial. It allows the cell to respond quickly
Respond quickly to what?
to changes in its environment, yeah,
Okay.
and fine-tune protein function.
So how does that happen? What are some of the ways?
How does that happen? Well, let's start with one of the big ones,
Okay.
ubiquitination.
Ubiquitination. Now that sounds familiar.
It does, doesn't it?
Didn't we come across that term earlier
We did.
when we were talking about mRNA degradation?
That's right.
It seems strange that the same molecule
The same molecule, ubiquitin.
could be involved in regulating both mRNA and protein.
Well, ubiquitin is a multitasker, you see.
Okay, a molecular multitask.
Exactly. A very versatile molecule.
So how does this ubiquitination process actually work
Work how?
when it comes to proteins?
Oh, it's quite elegant, actually.
Elegant. I like that.
Think of ubiquitin like a molecular postal code.
Postal code. Okay.
It gets attached to a protein.
Attached to a protein. Okay.
Yeah. And acts as a signal, like a label,
A label for what?
directing the protein to specific locations.
Oh, like an address label?
Yes. Or even marking it for destruction.
Destruction.
Yep. By a cellular machine called the proteasome.
The proteasome, that's like...
That's the recycling center,
So ubiquitin can either tell a protein where to go
like a traffic cop.
or send it to the recycling bin.
Exactly. And this process,
This whole ubiquitination thing,
Yeah.
it must be important for regulating protein levels.
crucial, making sure damaged or misfolded proteins are removed.
Like quality control again.
Exactly. Before they can cause any trouble.
So ubiquitin is like a master regulator,
You could say that.
controlling both the lifespan and the location of proteins.
Quite a powerful little molecule.
It is. But I'm guessing ubiquitination isn't the only tool.
Oh no, there are many more.
in the cell's post-translational toolbox.
A whole toolbox full of modifications.
So what other modifications are we talking about?
Well, let's delve into another one that's critical for regulating protein activity.
Lay it on me. Phosphorylation.
Phosphorylation. Now, that rings a bell.
It should. We talked about it before.
We did, didn't we?
Oh,
When we were talking about kinases.
that's right. Kinases and phosphorylation go hand in hand. It is. Phosphorylation is basically adding a phosphate group
Okay, adding a phosphate group.
to a protein, and it's a really common modification.
Common and important, you said.
Super important. It's like adding this tiny little switch
A kinase switch, okay.
that can flip a protein's activity on or off.
So it can activate or deactivate a protein.
Exactly. Or even change its function completely.
Wow. That's powerful for such a small change.
It is, and there are so many different kinases.
Oh, yeah, the
They're
kinase.
the enzymes that attach those phosphate groups.
Right, and each kinase has...
Each one has its own specific targets.
So very specific.
Very specific, yeah. And then there are phosphatases.
Phosphatases, what do those do?
They remove the phosphate groups.
Ah, so it's a two-way street. Exactly. Kinases add them, phosphatases remove them.
It's like a constant dance.
A dance of phosphorylation and dephosphorylation.
And that's how the cell fine-tunes protein activity.
Precisely. It's an incredibly elegant system.
Elegant, I like that word. Speaking of elegant,
Yeah.
we have to talk about non-coding RNAs.
Oh, yes. The non-coding RNAs.
They play a big role in post-transcriptional control too, right?
Huge role. They were overlooked for so long.
I remember reading about that.
Reading about what?
How they were considered junk DNA for years.
Oh, yeah, the junk DNA myth.
Because they don't code for proteins.
Right, but now we know better.
We know they're not junk at all.
Not even close. They're like
What are they like?
the puppet masters of the cell.
Puppet masters. Okay, I'm intrigued.
They control gene expression from behind the scenes.
I'm also thinking about RNA interference.
RNAi? Oh, yeah, that was a huge discovery.
It was a game changer for sure.
Totally revolutionized our understanding of gene regulation.
Scientists realized that these small RNA molecules
Tiny molecules.
could actually silence specific genes.
Like turning off a gene with a tiny switch.
It blew my mind when I first learned about it.
It was a Nobel Prize-winning discovery.
That's right, Andrew Fire and Craig Mello.
Those guys, yeah. They figured it out.
Working with those tiny worms.
C. elegans? The nematode.
Right, and what's crazy is
What the crazy?
this mechanism is found everywhere.
Everywhere. Plants, animals,
So
fungi.
like a fundamental process of life.
Absolutely. A core mechanism for controlling gene expression.
So how does RNAi actually work? Can you break it down?
Okay, so think of it like this.
Okay.
The cell has this defense system, right?
So defense system.
And it's constantly on the lookout for threats.
Threats like what?
Viruses, for example.
Oh, this is okay.
Or rogue pieces of genetic material.
So it's like a cellular immune system.
Exactly. And when the cell encounters double-stranded RNA,
Double-stranded RNA, that's
that's a big red flag for the cell.
a red flag.
It's a sign that something might be wrong.
Interesting.
So the cell kicks into gear.
What does it do?
It activates this system to neutralize the threat.
Okay, I'm ready for the play-by-play.
So first, the cell chops up those long dsRNA molecules
Chops them up how?
with an enzyme called dicer.
Dicer.
Yep. It's like a pair of molecular scissors.
So dicer cuts the dsRNA into smaller pieces.
Smaller pieces called siRNAs,
siRNAs, those are...
small interfering RNAs.
Right, and those siRNAs are the weapons.
Exactly. They're the guided missiles.
No, I did missiles.
Each siRNA teams up with a protein complex called RISC,
RISC.
the RNA-induced silencing complex.
Okay, RISC. And what does RISC do?
RISC is the launching pad, basically.
The launching pad.
It uses the siRNA as a guide
A guide to do what?
to find and destroy matching mRNA molecules.
So it's a seek and destroy mission.
You got it? RISC finds the target mRNA.
Based on the siRNA sequence.
Exactly. And then it cleaves the mRNA.
Cleaves it, meaning?
Cuts it, destroys it.
Oh, wow. So that effectively silences the gene.
Precisely. No more mRNA, no more protein.
That's incredibly precise.
The cell is a master of precision.
But we were talking about non-coding RNAs in general.
We were, and siRNAs are just one type.
Right. There are also microRNAs, the miRNA.
Ah, the miRNAs, they're like the CERNAV's little cousins.
Usins. How so?
They're both involved in gene silencing.
Okay.
But miRNAs are encoded by the cell's own DNA.
So they're not from external sources like viruses.
Nope. They're made by the cell itself.
So they're more like internal regulator.
Exactly. Fine-tuning gene expression during development.
And other processes.
All sorts of cellular processes.
So they're part of the cell's own control panel.
That's a great way to put it, making sure things run smoothly.
And they work with RASC too, right?
just like the siRNAs. They guide RISC to target mRNAs.
But mRNAs often have a different approach, don't they?
They do. Instead of completely destroying the mRNA,
They do what?
they can just block its translation into protein.
So it's more like dimming the lights.
Dimming the lights, yeah.
Then turning them off completely.
Perfect analogy. It's all about fine-tuning.
And this allows mRNAs to regulate a wide range of genes.
A huge range, everything from development to immunity.
I read that mRNAs are even involved in cancer.
Oh yeah, they play a complex role in cancer.
They can act as both tumor suppressors and oncogenes.
It all depends on which genes they target.
So they can either keep cell growth in check,
or fuel it out of control.
it's like they have a dual personality.
It's fascinating, isn't it? This duality.
Totally. Understanding how mRNAs work
And which genes they target.
could be crucial for developing new therapies.
Absolutely. It's a hot area of research.
So we've got these powerful silencing mechanisms.
RNAi and miRNAs, yeah.
But are proteins just sitting ducks in this whole process?
That's a good question. It's not a one-sided battle.
So there's pushback from the proteins.
You could say that. Cells have ways to counteract silencing.
Why would they want to do that?
Well, silencing is important, but so is expression.
You need both.
Exactly. Some essential genes need to be expressed.
Makes sense.
So there's this constant tug of war.
Between silencing and anti-silencing.
You got it. It's like an arms race.
A molecular arms race. Okay.
Some proteins can interfere with the RNAi machinery.
How do they do that?
Well, they can prevent siRNAs from binding to RASC.
So they block the silencing complex.
Exactly. Or they can inhibit RNAC's activity directly.
So they disarm the weapon.
You could say that. And some proteins even modify the mRNAs.
Modify the mRNAs. How?
They make them less susceptible to silencing.
So it's like a constant back and forth.
A never-ending battle for control.
It's like a cellular spy thriller.
With double agents and secret missions.
I love it. But with all this talk about silencing and anti-silencing,
Yeah.
it makes me wonder about this whole idea of genetic determinism.
Ah, the nature versus nurture debate.
Right. Are we really just puppets of our DNA?
It's more complicated than that, isn't it?
I think so. We've gone from this simple model
The RNA makes protein model
to this incredibly complex system.
with all these checks and balances.
Feedback loops, layers of regulation. It's mind-boggling.
It really is. So it's not just about the genes we inherit.
It's about how those genes are expressed
And modified. And controlled.
throughout our lives. Yeah.
That has huge implications for how we think about health.
And disease. It's not just about sequencing our genomes.
We need to understand how those genes are actually behaving.
In different tissues, at different times, under different conditions.
It's like we've been given this incredibly intricate tapestry.
A beautiful tapestry.
But we're only just beginning to understand how it's woven.
We're still unraveling the threads.
And that's what makes this field so exciting.
Absolutely. There's so much more to discover.
Well, before we get too carried away with the excitement,
Okay.
I want to go back to those post-translational modifications.
The ones we were talking about earlier.
Right, the ones that happen to proteins after they're made.
The finishing touches.
Exactly. It seems like even after all that regulation at the RNA level,
All that splicing and degradation.
the cell is still fine-tuning its protein workforce. Always fine-tuning. It's all about precision.
So can you remind us again
Remind you of what?
how cells tweak their proteins after they're already assembled?
Oh, sure. We were talking about ubiquitination.
Ubiquitination, right?
It's a key player in protein regulation. It's basically like adding a phosphate group to a protein.
Adding a phosphate group to a protein, right? We talked about that.
We did. It's one of the most common modifications.
And one of the most important, you said.
Super important. It's like adding this tiny, like a tiny little molecular switch
Yeah, molecular switch.
that can flip a protein's activity on or off.
Or even change its function completely.
Exactly. It can do all sorts of things.
So those kinases we were talking about,
Yes, the kinases.
they're the ones attaching those phosphate groups.
That's right. They're the ones doing the attaching.
And there's a whole family of kinases.
Oh, yeah. Whole bunch of them, each with its own specific targets.
They're very specific.
Super specific.
Yeah.
And then, of course, there are the phosphatases.
The phosphatases. Right, those are the ones that...
You remove the phosphate groups.
So it's like a two-way street.
Exactly. Kinases add, phosphatases remove.
It's a constant dance.
A delicate balance, you could say.
And all this to fine-tune protein activity.
Precisely. It's how the cell makes sure everything's working just right.
Can you give us an example of how this actually plays out?
Oh, sure. Let's talk about P53.
P53, that's...
It's often called the guardian of the genome.
Okay, the guardian of the genome. What does that even mean?
Well, it basically protects cells from DNA damage.
DNA damage. That's bad, right?
Very bad. It can lead to all sorts of problems, including cancer.
So P53 is like a superhero,
You could say that. It's constantly on patrol.
making sure the DNA is safe.
Exactly. And when it detects damage,
What happens?
it can trigger a number of responses.
Like what?
It can halt the cell cycle
Halt the cell cycle. Why would it do that?
to give the cell time to repair the damage.
That makes sense.
Or if the damage is too severe,
Then what?
it could even initiate cell death.
Wow, so it can either fix the problem or eliminate the cell.
Exactly. It's a powerful protein.
But if T53 is so powerful,
Yeah,
how do we prevent it from going overboard?
that's a good question. We don't want it destroying healthy cells.
So there must be a control mechanism.
There is. And it involves our old friend, ubiquitination.
Ubiquitination again. I thought we were done with that.
Ubiquitin is a busy molecule. It does a lot of different things.
Okay, so how does it control P53?
Well, remember how we said ubiquitin can tag proteins for destruction?
Right. It's like a label that says send to the recycling center.
Exactly. And in normal, healthy cells,
Yeah.
P53 is kept in check by a ubiquitin ligase called MDM2.
MDM2. Okay. And what does MDM2 do?
It basically tags P53 for destruction.
So it keeps P53 levels low.
Exactly. Preventing it from accumulating and causing problems.
It's like a safety mechanism.
A very important safety mechanism.
But what happens when there actually is DNA damage?
Good question. When DNA damage occurs,
Yeah.
MDM2 itself gets phosphorylated.
Phosphorylated. So that tiny molecular switch we were talking about.
Exactly. And when MDM2 gets phosphorylated,
What happens?
it can't bind to P53 as well.
So it can't tag it for destruction.
Exactly. Which means P53 is free to do its job.
To protect the cell from damage.
Precisely. It's a beautiful example of how these modifications work together.
It's like a cellular relay race.
A relay race, I like that. Passing the baton to ensure the right response.
And it all comes back to this idea of our cells as incredibly complex systems
Constantly monitoring and adjusting to maintain balance.
and protect themselves from harm.
Exactly. It's a remarkable feat of engineering.
You know, we started this deep dive talking about DNA as a blueprint.
Right, the blueprint for life.
But the more we learn about post-transcriptional regulation,
All these intricate
the
mechanisms.
more it seems like our genetic information is more like a
Like a what?
dynamic script.
A script that's constantly being rewritten.
Exactly. It's not just about the genes we inherit.
It's about how those genes are expressed and controlled.
And that has huge implications for how we think about health and disease.
Absolutely. It's not just about our genes, but how they're behaving.
It's like we've been given this incredibly intricate tapestry,
A beautiful, complex tapestry.
where we're only just beginning to understand the threads.
We're still unraveling the mystery.
Which makes this field so exciting.
Absolutely. There's so much more to learn.
Well, on that note, I think it's time to wrap up our deep dive.
Into the fascinating world of post-transcriptional regulation.
It's been an incredible journey, and I feel like we've only scratched the surface.
Just the tip of the iceberg, really.
Hopefully, it's given our listeners a new appreciation.
For the complexity and beauty of the molecular world.
And maybe even sparked a lifelong fascination with molecular biology.
That would be fantastic.
So until next time, keep exploring, keep questioning, and keep diving deep.
Dive deep.